Welcome, fellow clinicians, researchers, MDs, and PhDs, to this explainer. Look, if you're tuning in today, you already know perfectly well that aging isn't just some simple wear and tear process, right? Today, we're going to completely decode the intricate molecular cascades of systemic biological breakdown. We are finally shifting away from that fatalistic acceptance of chronological decline. Instead, we're mapping out aging as what it truly is, a highly complex, interconnected, and fundamentally targetable pathology. Okay, let's dive right into this. Our roadmap today covers 5 key areas. First, the evolutionary etiology of aging. Second, the pathobiology feedback loop. Third, multi-omic diagnostic clocks. Fourth, interventions and targeted therapeutics. And finally, the neurocognitive frontier. To truly grasp the sheer magnitude of where this field is headed, we first really need to recognize the profound human drive to conquer aging. Sigmund Freud once argued that on a fundamental subconscious level, humans simply do not accept their own mortality. And, for millennia, from ancient Egyptian mummification to the alchemy of the 1500s, this desire was just seen as foolish vanity. But today, modern mechanistic biology is finally turning this ancient psychological denial into a highly solvable, tangible engineering challenge. And the foundational proof that we are, in fact, dealing with an engineering challenge is this exact number right here, 120. According to contemporary research, this represents the theoretical limit, literally the absolute hardware ceiling of the current human lifespan. Now, for the clinicians listening, this number is absolutely critical. Why? Because it proves that aging is not some universal law of physics. We see organisms out there like the naked mole rat that barely age at all. This 120-year mark is simply a biological limit. It's a software design error that we can study, understand, and potentially surpass. Section 1, Evolutionary Etiology of Aging, the Resource Allocation Dilemma. Before we can reverse engineer the biological clock, we actually have to understand how it was built in the first place. And the truth is, evolution is a pretty terrible long-term engineer. It strictly prioritizes early reproductive fitness over late-life somatic maintenance. This evolutionary bias gives us the concept of antagonistic pleiotropy. Basically, the exact same genetic selections that make a 20-year-old fiercely resilient actually drive harmful physiological cascades in an 80-year-old. Think of it like over-engineering a race car engine for the first 100 miles, fully knowing it's going to literally melt the exhaust system by mile 200. That melted exhaust, that's an evolutionary oversight, and it is the direct root of the clinical inflammaging you encounter on rounds every single day. When we contrast these two biological imperatives side by side, we see the disposable soma theory in action. Historically, humans simply didn't make it to 80 in the wild. Consequently, our somatic maintenance, the heavy, metabolically expensive cellular repair mechanisms required to fix DNA, inevitably falls behind as we age. The biochemical budget just isn't allocated for long-term survival. Let's move to and see how this builds. Section 2, the pathobiology feedback loop systemic breakdown. So we're shifting from evolutionary theory into the actual self-amplifying molecular pathogenesis of aging. And let me be clear, this isn't just a random list of clinical symptoms on a chart. It is a vicious interconnected circuit. Environmental and metabolic factors drive genomic instability, which heavily accelerates telomere attrition. Once those protective caps hit a critical threshold, the cell slams the brakes and enters cellular senescence. But here's the catch. Instead of undergoing apoptosis like they should, these zombie cells just linger, and they secrete SASP, which is a toxic pro-inflammatory cocktail. This systemic inflammation triggers severe mitochondrial dysfunction, causing ATP to plummet and reactive oxygen species, or ROS, to spike wildly. And what does that amplified ROS do? It causes even more genomic damage, feeding right back into step 1. It is a complete runaway metabolic train. Section 3, Multi-omic diagnostic clocks, measuring biological time. Because aging operates as a closed pathobiology loop, playing clinical whack-a-mole with downstream symptoms, like, say, throwing antioxidants at ROS while ignoring the senescent cells completely, it totally fails. To break the circuit, we absolutely have to measure upstream biological age accurately. To understand this degradation, we look at the epigenome. If your DNA is the music on a CD, your epigenome is the laser reading it. Over time, epigenetic noise accumulates, much like scratches on that disc. The DNA hardware remains perfectly intact, but the software, it becomes completely unreadable. Cells literally forget their lineage-specific identity, leading to cellular mosaicism, where adjacent cells diverge genetically, creating profound tissue dysfunction. The contrast between our past and our future paradigms is incredibly stark. We are finally moving away from the old binary paradigm of just looking at chronological age and relying on reactive symptom management. By utilizing multi-omic diagnostics and site-specific DNA methylation clocks, clinicians can now track a patient's true biological age. This precise metric is a real game changer because it allows us to identify a highly measurable therapeutic window, giving us the exact leverage we need for proactive root cause interventions. Section 4, Interventions and Targeted Therapeutics, Applying the Patch. Now that we have the diagnostic infrastructure to actually map the damage, let's transition to the practical clinical applications and the biochemical tools we now possess to pry that therapeutic window wide open. Before we even get to experimental therapies, we have to acknowledge this incredibly empowering data point from the source material. Ready? Genetics account for only about 7% of human longevity. 7%. That means an overwhelming 93% is dictated by how you manage the environmental hardware. For researchers and practitioners, this effectively obliterates genetic fatalism. It drastically elevates the importance of proactive, daily clinical protocols. Managing that hardware starts with the actionable neuro protocol. Nutrition, exercise, unwinding stress, restoring through sleep, and optimizing cognitive engagement. I really want to emphasize to the medical professionals tuning in today that simple baseline inputs, like a 25-minute brisk walk to induce AMPK activation or prioritizing deep sleep to trigger the glymphatic system's clearance of amyloid beta, these physically modulate the cellular environment far better than any unproven billionaire serum currently being whispered about. Absolutely crucial to keep in mind. Now, what's really interesting about this timeline is how we are moving through escalating tiers of clinical intervention. We start with baseline modulators today, literally tricking the cell into autophagy through caloric restriction and exercise. But we rapidly escalate to pharmacological targeting, using mTOR inhibitors like rapamycin to pause cellular construction and force cleanup, alongside senolytics that actively hunt down those toxic zombie cells we talked about earlier. And looking ahead, by 2040, the translational pipeline moves squarely into advanced genomic engineering and actual in vivo epigenetic resets. Which brings us to the absolute bleeding edge of biogerontology, transient epigenetic reprogramming. This uses Yamanaka factors to literally wipe the scratches right off the epigenetic CD. However, the critical translational bottleneck here is control. You must pulse these factors just long enough to erase the aging marks without making, say, a cardiac cell forget it's supposed to beat, which would trigger disastrous teratomas. So, to safely bypass the ethical constraints of human trials, researchers are accelerating this exact discovery pipeline right now using AI-driven prediction models and ex vivo organ-on-a-chip microphysiological simulations. It's fascinating stuff. Section 5, the neurocognitive frontier, charting the unknown. As we solve these molecular bottlenecks, we really must pivot to the final neurocognitive implications of radically extending the human health span. This frontier brilliantly illustrates how deeply our environment interacts with our genetics. Take the APOE gene, for instance. The ancient APOE4 variant carries a drastically higher risk for Alzheimer's under modern metabolic stress compared to the newer APOE3 variant. But here is the critical clinical takeaway. Even with a highly penetrant genetic profile, over 60% of cognitive decline is entirely preventable via proactive lifestyle choices decades before symptoms ever appear. Your cognitive destiny is absolutely not strictly written in your genes. So the crucial point is, ultimately, our target state is the compression of morbidity. Longevity biotech is absolutely not about adding 20 years of frailty or maintaining patients indefinitely in nursing homes. No way. It is about fundamentally preventing the gradual decay of biological resilience. The goal is to maintain physiological vitality perfectly flat, near 100%, until the absolute chronological end of the human machine. But as we successfully transition from passively observing decay to actively engineering cellular resilience, I want to leave you, my fellow researchers and practitioners, with this profound, lingering question. If advanced epigenetic therapeutics successfully maintain a metabolically pristine body for 120 or even 150 years, what exactly happens to the psychological plasticity of the human brain? Do our neural networks become so deeply carved over a century that we hit a literal storage capacity limit? We may be fixing the biology of the cell, but we are just beginning to chart the totally uncharted reality of a radically extended human consciousness. Thank you so much for joining us for this explainer.
Thank you for being with us. Mega Psi is for you and for us.